So in this video, we're going to finish up talking about our introduction to biological sciences. So part 3 of lab 2 is using the metric system. And in the metric system, distance is measured in meters. Meters is the base unit. Now, the nice thing about the scientific metric system is that everything in the metric system is multiples of 10. So if I'm converting between meters to centimeters, 1 meter is equal to 100 centimeters. Meters to millimeters, 1 meter is 1,000 millimeters. Milli refers to 1,000. Meters to kilometers, 1,000 meters is equal to a kilometer. Millimeters to centimeters, we have 10 millimeters in 1 centimeter. And then if we look at millimeters to micrometers, we have 1 millimeter is equal to 1,000 micrometers. So this is just showing you some different representations. For me, I am a visual person, and so I like to see things in an image as opposed to just numbers. So the way that I think about the units is if you've ever seen what we call a yardstick, right? In the US, we use the imperial system, but in science and most of the world, they use the metric system. So what we call a yardstick is approximately the same distance as a meter stick. So if I were to look at a meter stick, the meter stick has all these numbers along it. And the meter is going to be 100 centimeters. So notice that this is 90, up to the end is going to be 100. So there are 100 centimeters in 1 meter. Now, you'll also notice that from, let's say, 6 to 7, that's our 1 centimeter mark. Between those centimeter marks, you see those littler lines. Those littler lines represent the millimeters. So 10 millimeters are equal to 1 centimeter. And you can see that down here as well. So we see this ruler and we see 10 millimeters are in 1 centimeter. If we look below, this is another representation. So 100 centimeters in 1 meter. And then lastly, if you think about kilometers, kilometers are very large distances. And so there are 1,000 meters. Think of a meter stick. There are 1,000 meter sticks in 1 kilometer. And just to give you an idea, if you think about how we talk about long distances in the US, we talk in terms of miles. Whereas if you went to Europe, they would be talking in terms of kilometers. And so if you're a runner, you probably know that if you run a 5K, for example, right, 5 kilometers is going to equal approximately 3 miles. So just to kind of give you an idea of scale as to how long these distances are, think of it visually. So again, a yardstick is approximately a meter. That meter contains 100 centimeters. So I just picture looking at a meter stick. You can see 90, 91, 92, 93. I just picture looking at a meter stick, and those whole numbers are centimeters. And I can picture that there's 100 centimeters on that meter stick. And then the little lines between the centimeters, those are the millimeters. So there are 10 millimeters in 1 centimeter. So let's talk about how we convert between units. The nice thing about the metric system is when we need to convert, let's say from centimeters to millimeters, it's a lot easier to do because it's all bases of 10. And so all it simply is moving the decimal point. If you think about the US system, right, when we talk about our measurements of length, our conversions are not as easy, right? We have 12 inches in a foot. We have 3 feet in a yard. The conversion between those units is not that easy. You actually have to multiply numbers out. In the metric system, it's simply just moving the decimal point. So let me walk you through how to convert between units. And once you get the hang of this, it actually is not hard at all. It is a common sense thing. So let's walk through it. So let's say I'm measuring the length of this juice box. So I have my juice box. The top is lined up with the 0 centimeters. So again, this is going to have centimeters. And so we've lined it up with the top of the ruler. And then we are measuring the length in centimeters. So if I were to look at this juice box and I look at where it is, it would be 13. Here's 0.5. Two more lines past that, so 13.7 centimeters. So 13.7 centimeters. So that's how many centimeters it is. Now we need to convert to millimeters. When you do that, you just need to cross cancel. So 13.7 centimeters. Now, if we want to convert to millimeters, you take the units you're trying to cancel out and they go on the bottom. So centimeters needs to go down on the bottom. And on the top is the unit we want to convert to. So millimeters is going to go on top. So now you need to think about the conversion between millimeters and centimeters. Remember that we said that 1 centimeter is equal to 10 millimeters, right? Those little lines represent millimeters. So there are 10 millimeters in 1 centimeter. So when we do the cross canceling, the centimeters, those units are going to cancel, and we're going to be left with millimeters. Now notice that the 10 is on the top. The 10 is on the top, which means we're taking 13.7 and we're multiplying by 10. So if I have 10 groups of 13.7, right? If I have 10 groups of 13.7, first thing you want to ask yourself is the number going to get larger or smaller? So if I have 10 groups of 13.7, my number is going to get larger because I have 10 times as much. So now all you have to do is you look and 10 has 1 0. What that means is we're going to move the decimal point 1 place. That's it. Move it 1 place because it only has 1 0. So now we want to think about, am I going to move my decimal point to the right or to the left? So I'm looking at my 13.7. Am I going to move it 1 place to the right, or am I going to move it 1 place to the left? If you think about it, if I move it 1 place to the left, my number gets smaller. If I move it 1 place to the right, my number gets larger. So again, that's where that common sense comes into play. If I'm multiplying by 10, I know that my number has to get bigger. So what that means is I have to move my decimal point to the right, and I just move it 1 place. So I make a little loop, and my decimal point is going to go there. 137 millimeters. So 13.7 centimeters is equal to 137 millimeters. The next one is asking how many micrometers. So for this, I'm going to start with millimeters because the conversion between millimeters to micrometers is easier. So I start with 137 millimeters. I need millimeters to go on the bottom because that's the unit we're trying to get rid of. And we're trying to get micrometers, so that's going to be at the top. So now we need to plug in our numbers. Micrometers, micro are very small. So if you think about cells, for example, cells are tiny. They are often microscopic. You need to use a microscope. So obviously a micrometer is going to be smaller than a millimeter. And so the conversion is going to be that 1 millimeter is equal to 1,000 micrometers. So notice we're going to cross out our millimeters. We have 137 times 1,000 because the number's on the top. So 137 times 1,000. Is my number going to get larger or smaller? Answer is if I'm multiplying by 1,000, it's going to get larger. So I'm going to write this down here just so I have a little more space. 137 has a decimal point here. How many zeros are in 1,000? 3. So I need to move my decimal point 3 places. Am I going to move my decimal point 3 places to the right or 3 places to the left? Well, my number is going to get larger, so I need to move it 3 places to the right. So I make myself loops to help keep track. 1, 2, 3. My decimal point's going to go here. Each one of these little slots gets a 0. So my answer is going to be 137,000 micrometers. So I end up with my answer as being 137,000 micrometers. So now let's convert to meters. For me, I'm going to go back to centimeters because I again can picture in my head that 1 meter is equal to 100 centimeters. So for me, that I can picture in my head, so I know that conversion. So we start with 13.7 centimeters. I already have centimeters, so they need to go on the bottom. Units I want need to go on the top. So again, 1 meter is equal to 100 centimeters. That meter stick has 100 centimeters on it. So now I'm taking 13.7 and I'm dividing by 100. So if I divide by 100, is my number going to get smaller or larger? If I divide it up into 100, my number is going to get smaller. How many places do I need to move my decimal point? I need to move it 2 because 100 has 2 zeros in it. So here's my decimal point, 1, 2, and my decimal point is going to go there. So my answer, 0.137 meters. Those are my conversions. So notice how much simpler converting units in the metric system is compared to converting units in our US system. We don't even have to do any math. It's simply just how many places do you move the decimal point and in which direction. If you're multiplying by the bigger number, you know that your number is going to get larger. If you're dividing by a number, you know that the number is going to get smaller. And so then you just move your decimal point in the appropriate direction. So for table 2.5, it's asking you to measure student height. Now, you might not have a meter stick at home, so we did this for you. So I stood up next to, this is actually a 2-meter stick. So it goes all the way to 200. And you can see that my height is right here. Now we gave you a close-up, so it makes it a little bit easier, but you need to record my height in centimeters. So my head goes right about here. So notice here's 176. So that means 175 is beneath here. So 175 point, and then come up with what you think for the decimal point. You're going to put that for your centimeters. This is measured in centimeters, so you need to convert between centimeters and meters. So do your conversion and then write my height in table 2.5 in meters. In the metric system, weight is measured in grams. Typical conversions, so again, if you think about the base unit being grams, those prefixes, the part that comes before gram, is consistent whether you're talking about weight, volume, distance, etc. And what I mean by that is if we convert from grams to kilograms, we have 1,000 grams is equal to 1 kilogram. Kilo again means 1,000. Grams to milligrams, we have 1 gram is equal to 1,000 milligrams, so on and so forth. So that base unit, that milli, kilo, deci if you're going to do decigrams, etc., prefix is the same in the metric system. Think about again the US system in terms of weight. We measure things in pounds and ounces, etc. Again, converting between those units, not as easy. 1 pound is equal to 16 ounces, etc. So again, converting in the metric system is a much easier task. So weight, our base unit in weight, is going to be in grams. Volume in the metric system is measured in liters. So our base unit is going to be liters. Typical conversion for volume, liters to milliliters. 1 liter is equal to 1,000 milliliters. Now, in terms of pure water, I'm not talking about tap water because tap water is not pure water. But if I'm talking about pure water has a relationship that 1 milliliter of pure water is going to weigh 1 gram. So that volume, that 1 milliliter of pure water is going to equal 1 gram. Volume of solid objects is measured in cubic centimeters, so cc's, or liters, by measuring the volume of liquid that the object displaces. So for this part of the experiment, you're not just measuring height. Now we're going to measure volume, and we are going to look at both measuring water as well as measuring the volume of a rubber stopper. And so I have 2 videos to describe this experiment, and then I will go over the data with you. So in this experiment, we are going to measure water. And so what I have is I have a serological pipette, and this is a 10 milliliter serological pipette. And so if you look at the numbers actually go in reverse order. So starting with 9 on the bottom, going up to 0 at the top. So this is a 10 milliliter serological pipette. And so to measure 10 milliliters of water, we are going to measure up to the 0. And what you're going to see is that when we measure water, water has a meniscus, meaning it has a point where it dips. And we want where the meniscus is to be at the 0. So let's go ahead and measure our water. So I'm going to take my serological pipette and I'm going to stick it into the water. And I'm going to use the pump and use the little dial to draw up the water. So I want the meniscus, the bottom, to be at the 0. So it's set at 0. So now I have my 10 milliliters of water. And you can see that on the scale here, the scale, we had recorded the weight of the beaker by itself. So now we're going to add the water to the beaker, trying to get all of the liquid out of the beaker, or out of the serological pipette. And we're going to put this on the scale, and we're going to re-record the weight of the water and the beaker. So now you're going to fill in this data in table 2-6. So notice that there's a place to put the weight of the beaker alone, and you can see the picture on the left. The beaker on its own has a weight of 49.65 grams. Then when the 10 milliliters of water was added to the beaker, now the weight has gone up to 59.26. So you're going to record that in table 2-6. To get the weight of the water, we're going to take the final weight, so meaning the weight of the beaker with the water, and we're going to subtract out the weight of the beaker, and that will give us the weight of the water. Now, if we were actually doing this experiment in a lab, if I was trying to find out the weight of the water in lab, I wouldn't actually do it this way. If I were doing this experiment, the way I would actually do this is if you put that beaker on the scale and you push that 0 tare button down here, this one here, what that's going to do is that's going to make the scale read 0 so that it's already subtracting out the weight of the beaker. Then when I add the water, it's only going to tell me the weight of the water. But for the purpose of this, you're going to record the weight of the beaker, you're going to record the weight of the beaker with the water, and then you're going to subtract out from that the weight of the beaker, and that's going to give you the weight of the water. Notice we were trying to measure 10 milliliters of water. If we measure 10 milliliters of water, what would we expect its weight to equal? Well, remember that for water, 1 gram is equal to 1 milliliter. So if that's the case, then 10 milliliters of water should weigh 10 grams. And so this is going to see, did the water weigh 10 grams? If it did not, you're going to see in your summary questions, you're going to address why that weight might not have been 10 grams. And we'll talk about that more in a little bit. So in this experiment, we are going to measure the volume of the rubber stopper. And so what I have is I have a graduated cylinder, and I filled the graduated cylinder to 30 milliliters. So what I'm going to do is I'm going to drop this rubber stopper into the water, and to measure the volume of the rubber stopper, I'm going to measure the amount of water that is displaced. So watch what I mean. So I drop in my rubber stopper, and you'll notice that the line has gone up, which means that the volume that has been displaced, meaning the amount that it moved up, is equal to the volume of the rubber stopper. So this is the data for the measuring volume experiment. So this is for table 2-7. And the first part asks for the water in the graduated cylinder, so the volume in milliliters. So what you're going to do is you're going to look at this graduated cylinder, and you want to find out what volume the water started at. So you're going to record the volume of the water. Then you have the water and the rubber stopper. So notice that when we added the rubber stopper went in and the volume went up. So we have 30 right here. We have 40 here. So you need to count how many milliliters is in this graduated cylinder. So you want to measure the volume in the graduated cylinder for the water and the rubber stopper. And you're going to record that in table 2-7. Now, the last row says final volume minus the initial. So you're going to take the volume of the water and the rubber stopper and subtract out the volume of the water. And what that's going to leave you with is the volume of the rubber stopper. And so you can see the rubber stopper displaced that water. You've probably noticed this before if you've ever taken a bath, for example. Let's say you're trying to run your bath and fill up the bath water. One of the things you might know is if you accidentally fill it too much and you go to sit in the bath after that, your body volume when you sit in that water is going to cause the water to rise. And you've probably experienced that at some point. And it gets to the point where if you filled it too much and you get in and your volume is big, that could make the water overflow because your volume is displacing that water. So that's showing that we can measure the volume of the rubber stopper by the amount of water that it's displaced. So when talking about temperature, the set points for water using the Fahrenheit scale. So again, this is the one we use more commonly in the US. For freezing, water freezes at 32 degrees Fahrenheit. Water boils at 212 degrees Fahrenheit. Notice when we use the metric system, and the metric system uses Celsius. It freezes at 0 degrees Celsius. Water boils at 100 degrees Celsius. Notice how much easier it is to memorize the set points for the Celsius scale compared to Fahrenheit. Freezes at 0, boils at 100. Fahrenheit is a lot more difficult, 32 versus 212. In this part of the experiment, you are going to measure the temperature of the classroom as well as the outside temperature. So this data is going to go in table 2-8, and this thermometer is in degrees Celsius. So you are going to record, the one on the left is the temperature in the lab room. The one on the right is outside on a June afternoon. And so you're going to record those temperatures in table 2-8. Now, if we need to convert between Fahrenheit and Celsius, there are several conversions that can be used. So degrees Fahrenheit is equal to 9/5 times the degrees Celsius plus 32. Or degrees Celsius is equal to 5/9 multiplied by degrees Fahrenheit minus 32. So let's practice a question. So in this question, I'm asking you to convert 78 degrees Fahrenheit to degrees Celsius. So I already have the degrees Fahrenheit and I want Celsius. So I'm going to use this bottom equation. So this is going to be equal to, so degrees Celsius is equal to 5/9. And then we have 78, that's my degrees Fahrenheit, minus 32. So we need to solve inside the parentheses first. So this is equal to 5/9 times 46, which is equal to 0.555 times 46, because 5/9, so if you divide 5 by 9, you get 0.555. So 0.555 times 46, or equal to 25.5 degrees Celsius. So if the temperature was 78 degrees Fahrenheit, in degrees Celsius, that would be 25.5 degrees Celsius. And so this is how you convert between temperature units. So here are some common conversions. You do not need to memorize this. It's just for you to get an idea of scale. So 1 foot is equal to 30 centimeters. 1 yard is approximately equal to 1 meter. 1 mile is equal to 1,600 meters or 1.6 kilometers. 16 fluid ounces is equal to a pint, which is equal to 500 milliliters. 2.2 pounds is equal to 1 kilogram. Human body temperature, 98.6 or 98.7 approximately, is equal to 37 degrees Celsius. So human body temperature in degrees Celsius is about 37 degrees. So part 4 of this lab is looking at graphing. And you're going to be doing several graphs throughout this class, and you need to understand how to set up a good graph. So there are several keys to a good graph. The first component is that you include a title at the top that clearly indicates the contents and the purpose of the tables or graph. So it's basically summarizing what the graph shows. You want to label all relevant parts. So any rows or columns or lines or the x-axis or the y-axis, you'll see this in a minute when I go through an example. You want to make sure to label all measurement units. Millimeters, centimeters, milliliters, minutes, degrees Celsius, etc. Right? Because if I write on my graph just time, well, that time, just writing time is not very descriptive. Are we talking in terms of seconds? Are we talking in terms of months, days, hours? What is my unit of time? That makes a big difference. So you always have to make sure that if there are units, that you include those units on your graph. You should scale your axes to accommodate all your data. No data point should exceed the values defined in your axes, and the spacing for the scale should be even. So you want to, before you even put numbers on your graph, you always want to look at your total data and see what is your highest number, what is your lowest number, and then make your axes accordingly so that it will all fit within your graph. Because let's say you're counting by tens and your highest data point is 150, but you only made your graph go to 100, well, 150 is going to be off of your graph paper. That's not going to work. So always look at your data first, look at your numbers, and then decide on your scale. You have to make sure that when you're doing your scale, that you're counting equally. So like for the main lines, if we do 10, 20, 30, 40, 50, each of those gaps, that space between them, is going to be an equal amount. So I can't have my scale like one mark has 1, and then the next one is at 3, and then the next one is at 15, and the next one's at 17. It doesn't work that way. You have to have your scale with even spaces, and you'll see what I mean by that in just a minute. Draw your graph neatly and accurately using straight lines where appropriate. So if you need to, do a line graph, you might want to use a ruler to make your lines straight. So when we do an experiment, we have to understand the difference between the independent variable and the dependent variable. The independent variable is the variable that you are manipulating. The dependent variable is the variable that you're testing the response for. So let me walk you through an example so that you can see what I mean by this, because those two definitions can be a little bit tricky. So the first type of graph that we're going to talk about is what's called the bar graph. And a bar graph is used when data is grouped into ranges or categories. So let's say we wanted to graph the average height of males versus females. We wanted to see on average, are males taller or are females taller? So when we have our graph, our x-axis, our horizontal line is going to have our independent variable. Our y-axis or our vertical line is going to have our dependent variable on it. So here's my graph paper. So if I want to graph average height versus gender, meaning male or female, you have to ask yourself which one goes where. Does gender or average height go on the x-axis? So the easiest way for me to figure out which one goes where is I ask myself, which one depends on the other? So does average height depend on gender? Does gender depend on height? So can your gender change depending on your height? Answer is no. That statement doesn't make any sense. But the average height is dependent on the gender. So the average height is the dependent. That's what's going to go on the y-axis. And gender is the independent. That can't change. We're measuring the response for the average height, but the gender is going to be independent. So here we have height is going to be our dependent variable, right? The average height is going to depend on gender. Notice that I have my units on there, so my height is in inches. Here is my scale. So the data that's going to be placed on this graph is going to be made up data, but it's just to illustrate the point on how to make a strong graph. So notice that on my scale, I have 15, 30, 45, 60, 75, etc. So notice that my scale is counting by 15 each time. And so they're evenly spaced and they're set up appropriately. Then we have male and female down here, and we need to label that axis as well. So we're going to label that gender. And so now we just need to put our data on our graph. And so let's say on average that males are 5 foot 10 inches. Let's say that they're 70 inches on average. So we put males at 70, and let's say that females on average are 5'4". So maybe we have our graph at 64 inches. And so this is our graph. This is a bar graph because it's distinct groups, males versus females. We're not measuring something over time. We're measuring distinct groups. We have two populations. We have male and we have female. So we have our axes labeled. We have our scale on there, but we're still missing something on this graph. And what we're missing on the graph is going to be the title. So the title for this could be mean height by gender. You could also have the title be on average, males are taller than females. Something along those lines. You want your title to be descriptive. So you shouldn't ever just say height versus gender. That's not descriptive enough. So just don't use versus in your title. That should be not used when possible. So this is another example just showing you bar graph. So what you can see is on the x-axis we have blood types, and there are 4 blood types, A, B, AB, and O, and we have percentage of the population. And so what we're basically doing is in this graph, we're comparing the percentage of people in country A that have type A blood versus the percentage in country B versus the percentage in country C. So is this graph set up properly with the right axes? Does the percentage of the population depend on the blood type? Or does blood type depend on the percentage of the population? Well, the blood type can't change depending on the percentage of the population. So that one's not true. It's the percentage of the population depends on the blood type, right? Depending on which blood type you're looking at, that's going to change the percentage of the population. So my dependent variable is my percentage of the population. My independent is going to be the blood type. Notice that for this bar graph, we have 3 sets of bars because we have country A, country B, and country C. So for this one, you need to have a key or a figure legend. And that figure legend is there basically to indicate what the different colors mean on the graph. That kind of lavender gray color represents country A, and the purple color represents country B, and the yellow color represents country C. And so what you can see if you look at this, for example, country C has more people with blood type A and B than country A and B. So country C has more A and B than either country A or B. Country C has the lowest percentage of O-type blood when compared to country A and B, etc. So what you can see is depending on what country you're in, that can change the percentage of the population that has a particular blood type. So this is a bar graph. It's used when data is grouped into ranges or categories, and they're distinct, right? Blood type A is different than B. B is different than AB, etc. So this is an example of a bar graph. So now let's look at a line graph. A line graph is used to summarize how two or more variables are related and how they vary depending on one another. Usually line graphs are used for continuous data. For example, if you're measuring something over time. So I gave you two examples here. So one of these graphs says coastal water temperatures. And in this graph, what they're doing is they're measuring the temperature of the water over a period of 5 years. So from 2000 to 2005. And they're looking for, did the coastal water temperature fluctuate over time? So we have 2000, 2001, 2002, etc. This can be a line graph because it's continuous. You would extrapolate that somewhere between 2000 and 2001, so during that year period, that the temperature would fall within this range. This is continuous data. We're measuring something over time. The graph on the right is looking at the levels of estradiol in women. So looking at basically estrogen precursors. So age is in years, estradiol concentrations. You want to make sure that they're on the right axes. So does the estradiol concentration depend on a woman's age? Or does a woman's age depend on her estradiol level? And the answer is estradiol levels depends on the age, right? As a woman approaches menopause, her estrogen levels are going to drop. So this is on the right axis. The estradiol is the dependent. It's on the y-axis. And the age or years is in the x-axis. And so what you can see is when they measured this, that as the women get older, their estradiol level drops. And again, that's because it drops during menopause. And so then it stabilizes. And so this is an example of a line graph. We're measuring something consistently over time. A pie chart is used to show the relative size or proportion of related pieces of information. So let's say, for example, that we are looking at the grade distribution in a class. So this is another class. So Biology 101 student grades. And so we have this pie chart and it's showing of the whole class, so of 100% of the whole class, what percentage had an A, what percentage had a B, C, D, F. Notice that this does a really nice job of looking at the proportion. So 9% of the class got an A, 21% got a B, 49% got a C, 15% got a D, and 6% got an F. And so if this is your proportion, you'll notice that almost half of the students, you can see it even visually, almost half of the students got a C in the class. A fairly low percentage of people in the class got a F or an A. So if there are 55 students in the class, how many of those students received an A grade? What you need to do is you need to take 55 times 9% is equal to 0.09. So if you take 9 over 100, 0.09. So now we have 55 times 0.09, which is equal to 4.95. But can we have 0.95 of a student? No. So we need to round. That means that approximately 5 students out of 55 got an A in the class. So pie charts are great to show the relative size or proportion in a population or in a class. A scatter plot is used to determine if a relationship exists between two measured variables. Bar graphs and line graphs are used when you know that one variable, the dependent, changes in accordance with the other variable, the independent, because they're somehow related to one another. A scatter plot is used if you do not know if the two variables are related and would like to determine if they are. So for example, if we wanted to see if there was a correlation between the number of hours that a student studied and their score on the exam, right? We want to know if you study more, do you get a higher grade? So on this graph, each of these little blue dots represents a student. So like this student here, they studied 20 hours a week, and that student got about a 95%. This student that got a perfect score maybe studied 18 hours. This student down here, they only studied for maybe 1 hour a week, and they got, a 35 on their exam, and so on and so forth. Now, when you do a scatter plot, you don't connect the dots. They're not continuous. They're distinct people, right? Each dot represents a person. So we don't connect the dots. Instead, we draw a line of best fit. We draw a line that goes in the middle of the data points. So you want that line to be in the middle of all those data points, and you want those dots to be as close to the line as you can possibly get them. Now, if you see your graph going up and to the right, that is what we call a positive relationship. And what that means is as the number of hours a student studies, that is going to have their score go up. So the more the students study, the higher their grade on the exam. It's not true for every student, right? But that there is a correlation. There is a correlation that the more you study, the higher your score. That's a positive relationship. Both are going up. A negative relationship would be when the line points down and to the right. So if I'm graphing this and my line goes this way, that would be a negative relationship. Because what that means is that as my hours go up, my score goes down. That's an inverse relationship or a negative relationship. So a negative relationship is where the line goes down and to the right. No relationship exists when the line is parallel to the x-axis. So if you had a line that goes just straight across, that means that there's no relationship, because that means that your points are all over. There is no correlation. There's no relationship between those two aspects. If we say that it's a strong relationship means that the points are very close to the best fit curve or fall directly on the line, meaning that is a very strong correlation because it's very close to that line. The weak points are a moderate distance from the line, but clearly show a linear pattern in which to draw the best fit line. So you know that it still should go up and to the right or down and to the right, but they're not as close to the line as a strong relationship would be. And so that's your scatter plot. So for your graph for this lab, what I have here is this is data from a previous semester. And so what the students did was they measured their hands. They measured their hand length, so from the base of their palm to the tip of their tallest finger, and they recorded their hand length in centimeters. And we are comparing males versus females. We want to see on average, do males or females have a greater hand length? So what we did was we had all the students in the class measure their hands, and then they brought their data up and we compiled class data. And so you're going to use this table to fill in the hand length data. So for the females, student number 1, so one of the students had a hand length of 17 centimeters. Another student had a hand length of 16.5 centimeters, and so on and so forth. And then the same was done for the males. Now, we have all the data for all the females and all the males. But what we want to know is what is the average hand length. So what you do for this data is you take, so for females, and you add up the hand lengths for all of the females in the class. So the sum in this case, the total, would be 251. To get the average, you have to take that sum and divide by the number of data points. So you would take the 251 and divide by 15. And so the calculated average for females for hand length is 16.7. For males, when you add up all those numbers, your sum is going to be 292.6. And when you divide that by 16, there's 16 data points, you get 18.3. So what you can see is on average, do males or females have a longer hand length? The answer is that the males on average have a greater hand length. But notice this is on average. It's not always the case. So if we look, we have a couple females who have a hand length of 18. And some of the males have a hand length of 16.4 or 16.2. So in some instances, the female does have a larger hand length than males. But on average, if we look at a bunch of males, on average, the males have a greater hand length than the females. And so what you're going to graph for this graph is you're going to measure the hand length, the average hand length for males versus females. So you need to make a graph based on that data. So again, on your x-axis needs to be your independent variable. My x-axis is going to be my independent variable. My y-axis is going to be my dependent variable. And so in my graph, right, I'm graphing average hand length and gender. So does average hand length depend on gender? Or does gender depend on average hand length? Gender can't depend on average hand length. We can't change gender. So gender is going to be independent, and the dependent is going to be the average hand length. Average hand length depends on gender. So we have our average hand length. Notice there's something missing though. What is missing from that label for the axis? Answer, units. Centimeters. The hand lengths were measured in centimeters. On the x-axis, we're going to put male and female, and we're going to have gender. What type of graph is this going to be? Answer is, this is going to be a bar graph because these are distinct groups, males versus females. So you need to look at your graph and look at your data, figure out the scale that you're going to use on your average hand length, and then make a bar. So you're going to make your bar and then shade it in for males versus females. And so that's what you're going to do for your graph. What else is missing from this graph? So once I put my data on there, I'm just making it up for now. Once I put my bars on there, what is still missing from my graph? Answer, title. So you have to come up with the title yourself. Don't forget your title. Title, exclamation point. So now, looking at our summary questions, I want to give you a little bit of feedback as to how to answer your summary questions. Question number 6, based on your data from table 2-6, did the 10 milliliters of pure water weigh exactly 10 grams? Briefly explain why or why not. So you want to make sure and explain with data. So if you said that the 10 milliliters of water did not weigh 10 grams, you should also include in there, how much did it actually weigh? So if it didn't weigh 10 grams, how much did it weigh? So that's the first thing. So explain with data. If it did not measure 10 grams. So if you said that it wasn't accurate, it wasn't 10 grams, I want you to come up with 2 reasons, not just one reason that it didn't weigh 10 grams, 2 reasons. So what you're looking for is you're looking for 2 things that might have made it so that the water did not weigh 10 grams. Now, you cannot simply say human error. What type of human error? Be more specific. So don't just say human error. What specifically could have done? One thing you want to know about water is that when you're measuring water, if this is, let's say, my serological pipette or even my graduated cylinder, and here's my 10, so that's my 10 milliliter mark, water forms what's called a meniscus. That is that it goes down. It doesn't just go straight across. When we measure water in any type of vessel, whether it be a beaker or a graduated cylinder or a serological pipette, you always want to get at eye level and measure the bottom of the meniscus at the number you're trying to get to. So for my 10 milliliters of water, I needed to make sure that I was at eye level and that the bottom of the meniscus was at the 10 mark. So that's just something to keep in mind. If you were actually doing this in person, whenever you measure water, you have to get at eye level to where the water is and make sure that the bottom of the meniscus is at the value that you're trying to get at. So come up with 2 reasons if the water did not weigh exactly 10 grams. Where might some error have come into play? What might have caused not enough waters, right? So if your number was less than 10, that means you lost water along the way. Where might you have lost some water, etc. Question number 7, convert your body temperature from degrees Celsius to degrees Fahrenheit. If your body temperature is 40 degrees Celsius, would you be concerned? Explain why you would be concerned. Use data. Show your step-by-step calculation below for credit. So under this little lines part where you're going to write your answer, underneath that, show the calculations. So take that 40 degrees Celsius and convert it into Fahrenheit. And then you want to see, based on what number you get, should you be concerned? And so your answer is going to include what the conversion is and why you would be concerned. And so that's for number 7. Quiz 2 study guide. You need to know the parts of the atom. So the protons, the neutrons, the electrons. Protons are positive, neutrons are neutral, electrons are negative. You need to know how to determine the number of protons, electrons, and neutrons and the molecular weight. So if you were looking at, let's say, that periodic table like we did in the practice, you should be able to tell me how many protons, electrons, and neutrons it would have. What happens to an atom's charge when it loses or gains an electron? The difference between a base, an acid, and a buffer. Understand the metric system and how to determine the volume of a solid object in milliliters, right? By looking at the volume of water that was displaced. And include the relationship of the weight and the volume of the water. So you should know that water, for example, 1 gram is equal to 1 milliliter for pure water. Be able to convert between units in the metric system. So I would give you the conversion, meaning I would tell you that 1 meter is equal to 100 centimeters, but then you have to apply that to do the calculation. You won't use a calculator because for that, you don't need a calculator. You're simply moving the decimal point. So you don't need a calculator. If given the equation, be able to convert degrees Fahrenheit to degrees Celsius and vice versa. And that would be if I gave you a calculator. Determine what type of graph to use if given a scenario. So if I gave you a particular situation, I could ask you what type of graph you would use. And so you want to understand, why do you use a bar graph versus a line graph versus a scatter plot, for example. Determine independent and dependent variables and know which one goes on the X or the Y axis. So you should be able to identify which is the independent variable, which is the dependent, and know that the independent goes on the X and the dependent goes on the Y. So 7 questions come from that study guide. 3 questions come from the pre-lab. Read through lab 3 before you take quiz 2, because 3 questions will come from lab 3. So you should define the terms at the beginning of the lab, do that before the quiz, and you need to read through the lab ahead of time so that you understand what you're doing. If we were doing this in person, that would be something you would have to do. You wouldn't have a video ahead of time. You would actually be reading through the lab ahead of time. And the purpose of that is so that when you come in and do the lab, you already have a general understanding of what we're doing that day. It's always a good idea to read through labs before you actually do them. And so that's why we include those pre-lab questions. You want to read through the lab and have a general understanding of what it is we're doing that day.